All right, guys, let's go ahead and get started here. Picture blank. Yes? No. Why must you always switch things on my computer? Swap. There we go. Okay. Okay, real quick thing. Dr. Gill asked me to share this with you guys, even though it's after the date, because obviously today's not 26. There's a SCANUS thing, which is basically the Society for Advancement of Chicanos and Hispanics, Native Americans, and STEM. This is an organization that's actually recently started at Baylor. Highly would like to promote this just because it's an area where we can focus kind of helping underserved communities and underrepresented communities within the science field, especially in biology. If you guys are interested in this, you don't have to be, Hispanic or Latino or Native American to join this. This is just an open community thing, helping to kind of promote this. If you would like more information, definitely talk to, well, there's two places. You can email scanus.baylor@gmail, or you can talk to Dr. Corinne Gill. She's very, very nice. But they have lots of information on this, what have you. So I thought, just so that you guys are aware of that and you have that available, so what have you. Let's talk a little bit about certain things. Okay, lab practical review worksheet. I know you guys had this there. Hopefully you guys have started working on this. What was that? Can we work on it? Sure, I was going to go over some of the things on here. The whole thing, I don't know. How many questions did I put on here? Gosh. We'll go over a bunch of them. Ooh, okay, so this is actually a big one. I never changed this one. I'm sorry, guys. This is obviously not the same kind of test that we use. This is the old style test. I forgot that was on there. But yeah, we can go over a bunch of things. And then I was going to quiz you guys on a couple of questions that are just stolen from last semester but aren't going to be present on this semester in the same format, so you guys can see how those would look, okay? All right, so first thing up, rapid tests. The rapid tests used in this lab are immunosorbent assays. So immunosorbent, they stick to the thing, immunologically speaking. So rapid test for blank, and it looks for blank antigens. Blank with a dye attached to the antigen, and then blank embedded on the test strip, also bind the antigens. Okay, so does anybody know which is the rapid test that tests for blank, and it looks for blank antigens? So it's got these embedded on the test strip. Does anybody know what that one is? Huh? Yeah, it was this. It was the Streptococcus test. So Streptococcus pyogenes, it was the throat swab style test, and then you stuck it in the liquid, and then it sucked it up and went up the test strip. And it's looking for those strep A antigens. So we call them group A antigens or gas antigens. So that's an easy one to identify. So group A antigens, group A strep. And then we put in there antibodies that have a dye bound to them. And so basically, if the gas antigens are present as they go up the strip, the antibodies that are also in the solution will bind to the gas antigens, and then there's gas antigens bound in the strip. So if the gas antigens are bound to the antibody and then another antibody is bound to it that's labeled, you'll get a nice little pretty colored line. And then you have a control line. The control line actually binds the antibodies themselves as they're going along so that we can verify that the antibodies have actually properly migrated up. And so it binds the control line, which is a really, really important part of this. Now, if the control strip doesn't appear, then what do we say with this test? What does this test result? Invalid. That's a really important one. Now, real quick question, which comes first, the test line or the control line? Test line comes first. Why isn't the control line first? What was that? So that's a bit of it. So that's one thing. I know your TA has talked about that probably a little bit. It can reduce the number of antibodies coming through because the control line's grabbing them. But realistically, in all likelihood, there's way more antibodies than you'd ever want in any solution to get a fully saturated line on both lines. What's really happening is if the control line is in front of the test line, you don't know if the liquid has gone all the way up the test strip to the antibody line. You have no way of telling that. But if you have the control line on the other side of the test line, you know that if the control line pops positive, then that means that you've definitely had antibodies go across the test strip. And so that's why we always put the control line on the other side, because that's an insurance that the antibodies have definitely gone all the way across the test strip. So that's the reason why. So that's the real reason why. So, and this is one of those things where it's like, yes, so the TA, that explanation is correct. You can have a reduction in the number of antibodies present. That's not true for this particular test for strep because we just load it up with tons. But some of the other tests that have like the ability to give you a range of how positive it is can exist. Yes? Yeah, so the actual test, let's see here. So the actual test line and the control line on the strip, right? They're both very similar because the test line has an antibody embedded on it and the control line has an antibody embedded on it. The difference between these is what they bind, and the control one binds IgG, which is an antibody, and this one binds the gas antigens, so group A strep antigens. And what this one is doing is it's trying to bind any group A strep antigens so that they stick here, and that our test antibody that's got the colored dye on it, this guy, he'll come over and he'll stick right here. But this guy will also get stuck right here so that you will verify that this guy actually made it all the way up the test strip. And as I'm drawing this, I realize that I'm blocking your view with my thing, but yes. Does that make sense? Okay. Does that clear it up for everybody? I know that one was like one that people were having problems kind of grasping because it's not a common testing. Yeah, do you have a question?
Yeah, for number one, could you set
Amino what? So the whole test type, so we call it ELISA, which is enzyme-linked immunosorbent assay. Immunosorbent is what it is, so it's an immunosorbent assay. So that's what it is. So you guys should know what that whole definition is, was the enzyme-linked immunosorbent assay, ELISA. So you should know that whole word. That's an important one to know. But if you just kind of broke it down to immunosorbent assay, I'd be fine with that. But immuno-based is not really, it's probably be okay, I'd probably take it. But immunosorbent assay is better. Okay. So the rapid test for blank blank uses beads. So which one is the bead test for? Which bacteria? Staphylococcus aureus, very good. Okay, now this one's a slightly different one. It uses latex beads, right? So latex beads are the key part of it. And then on top of that, they have two different things on them. They've got immunoglobulin, and then they've got fibrin on there, right? And so they've got two different things on here. And the reason why we have two different things is because the antibodies are not binding the staph. The antibodies are binding are being bound by the Staphylococcus's own ability to bind antibodies. So it uses this ability to bind its antibodies to kind of shield itself from the immune system. So staph kind of has that really unusual property, so it can bind that. And then the other part is the fibrin, and it's basically got a set of enzymes that cause fibrin. So if we're looking at the latex bead, it's got the antibodies on it, right? And then it's got this weird fibrin thing. And staph will actually have enzymes that convert this to the kind of interlinking mesh. And so basically all you're doing is you're just creating a big mesh of stuck together beads. And that's actually all you're visualizing on the test strip, is that coagulation event, that basically clumping up. We don't call it coagulation, it's agglutination, but that's what we're looking for. So In this case, the antibody isn't binding the staff is binding the antibody, which is weird. But that's kind of the trick for that one. Okay. Now, aside from this one, if I were to show you guys another bit, hold on one second. Okay. Okay, so that part clears up the amino-based one. That one's the one that people tend to forget because that was at the very beginning of the semester, but definitely go back and kind of look over that, kind of make some flashcards or something. Let's talk quickly about the unknown isolation, which is actually very short. You should remember what the 3 plates are for isolation. Does anybody remember what the 3 plates are that we used for isolation in the very beginning? TSA, McConkey's, and PEA. What does PEA select for? Gram-positives. Now, there's a kind of tricky part with this. Is PEA really, really good at selecting for gram-positives? Or is it kind of okay? Yours was great. Okay, good. So it is actually only kind of good. So it's mostly good at inhibiting. Sometimes you'll get really aggressive growth, but for the most part, it works pretty okay. The McConkey's auger, on the other hand, is really good at selecting for only one type of organism because the dyes present in it are really, really good at saying, hey, do not allow anything else to grow on here other than gram-negative. And of course, we have TSA, and TSA lets anything grow on it for the most part pretty easily. But the benefit of having the TSA plate is that it doesn't have any dyes embedded into it. So for your McConkey's auger plate, you can't use McConkey's auger plate because it has dyes built into it to do your gram staining from, because you don't know if it's that color because of the dyes in the McConkey's auger or because of the gram stain. So you have to take it from the TSA. You can do it from the PEA because the PEA doesn't really affect that, but you have to do at least something from the TSA. So that's the very first important thing. For the gram staining, let's see here, what are we talking here? So you shouldn't use this media to do your gram stain because the dye's on it. In this case, that's the McConkey's. And we just talked about the PEA. Gram-pos. PEA is just selective. It's not differential and only allows gram-positive to grow on there. So that's another part. So PEA, unlike McConkey's, is only selective. McConkey's is also differential, so it allows you to differentiate if something's able to ferment lactose or not ferment lactose. PEA doesn't do that at all. It's just selective media. It's not differential. So it can't tell you anything about the chemical properties of what the bacteria can process. It just says, hey, are you gram-positive or gram-negative? Okay. Okay, if you want to get an colony, you should draw a pattern like this, which I left blank. Now, there's a couple of different ways you could do this that I would accept as correct, but the one that we talk about in class is that 3-way streak for isolation, which I know we've kind of talked about a whole bunch. The really key one for this drawing, though, is directionality, because directionality gives you, each time you do this step, you want to lower the number of bacteria so you get an isolated colony. So you always start from the far end like this, and if I were to draw this, it would be like this. Then you'd flame your loop, let it cool down. You're not going to put more bacteria on, you're just going to go drag it across. And so you're going from the lowest concentration and lowering it more each time. So that's the general gist of this kind of pattern that you want to go for. This last one, of course, you go from the lowest end here, and we'll drag it into this final quadrant, trying to spread it out. And notice how I don't go back and forth a whole bunch over here, because every time you cross this line into here, you're bringing more bacteria down. So you only want to bring bacteria down once. So you go across 2 or 3 lines, and that's it. But that would be the general pattern. So it's a pretty easy pattern, very simple, but it works really effectively. There's some other crazy patterns, and I'm sure you guys have seen them probably in your lab manuals, where it's like, oh, you can do like a bunch of streaks like this, and then a bunch of streaks like this, and then a bunch of streaks like this, and then like this, and then like that. That works too, but that's not very effective, especially if you're just learning, so just keep that in mind. That's why we don't use that in lab. Okay, let's take a look at this. Gram results. All right, so these are pretty easy. This is kind of representative of what I would give you guys. You look at this and you go, oh, hey, this guy right here, that's a red colored guy. So that's pretty easy. You go, yeah, that's got to be gram-negative, right? Gram-negative. But you should also know the shape and arrangement. In this case, the shape, a little bit confusing on the projector, but if you guys print it out, it's very clearly these kind of foci. They're not bacilli, so they're not elongated, so they're little dots. So we'd say, yeah, that would be foci. And the arrangement, what would you guys say this arrangement is? Strepto. Very good. So strepto arrangement. This guy, we say gram-positive or gram-negative? Who says gram-positive? Who says gram-negative? Who didn't vote? I saw a bunch of you guys. You guys can vote. It's okay. Nobody's going to judge you. I'm not going to call you out personally unless you don't vote. Okay, let's try that again. Who says gram-positive? Okay, who says gram-negative? Who still didn't vote? I can see one person who didn't vote right there. I watched you. Okay, I do pay attention sometimes. Yes, it is gram-positive. And for the person who didn't vote, what would you say this arrangement is? Person who didn't vote, what was this arrangement? Huh? It is not random, actually. It's got a consistent pattern. Do you have a guess? It is staph. Very good. It's staph because it's got that grape-like clusters. It's got those kind of little patterns where it's clustered together. If it were random, we would see a lot more white space and then individual bacteria by themselves. But because we see these kind of repeated patterns clumped up together, we'd say that that would be definitely Staphylo. And the shape is foci because you can't have Staphylococcus, but Staphylococci. All right, last one here. This one's really hard, actually. It's really hard in the fact that this is small, but if I blow it up like this, it's much easier to see. What is the arrangement for this guy? Tetrad, very good. This is actually really easy, and it's actually one of the most important tests because if you saw some tetrad bacteria on an unknown, that's only two organisms. That's Micrococcus luteus and Cochuria rosea, or Micrococcus roseus, depending on how old of a file you're looking at for identifications. But yeah, that's the only two tetrad bacteria that we have in the entire lab. So that one is actually really important for narrowing down this thing. Okay, there you go, very good. Yes? How do I know that one that I showed you guys was not Diplo? Yeah, very good question. So the general pattern that you say is, okay, what's likely to occur in nature? Pairs can happen pretty commonly in nature, but tetrad is just really not that common. And you can see, looking here, you can go, okay, well, here's a group of four, here's a group of four, here's a group of four, and you see that repeating pattern of four multiple times in a row. That is not just random chance. So you'd go, yeah, that has to be tetrad. So that's a very good question. Yeah, absolutely. All right, let's talk about gram-positives. The very first test we do is for which enzyme? Catalase. And what does catalase do? Okay, but did you know that because you were looking at your notes? Okay. That is true. Okay. If you happen to be catalase negative, what is your general oxygen preferences? What would you guys say to that? That's probably not in your notes. Yeah, slightly anaerobic. So you'd prefer, now they're not strict anaerobes because strict anaerobes in our class would not be helpful because oxygen is everywhere and it would kill them. So we use facultative anaerobes, but they're those that kind of lean towards the side of I'd rather not have oxygen. So all of our catalase negative are those organisms that grow much lighter in general. So if you guys remember, if you guys happen to have these, it would be things like Streptococcus pyogenes, Enterococcus faecalis, Streptococcus galactae, those kinds. They don't like as much oxygen because they don't have the enzymes to handle it as well. They do have other enzymes that handle it, but not as well. And catalase is one of the big enzymes that happen. In fact, it's the fastest enzymatic reaction known to man. It is really, really cool. So, let's see here. This enzyme converts hydrogen peroxide into oxygen and water, yeah. Okay, the two main groups of bacteria are catalase positive and catalase negative. You could also say that they are the Streptococcus and the Staphylococcus, even though they're not all Streptococcus and they're not all Staphylococcus, but we kind of just hand wave that aside. Yes, the Staphylococcus ones generally are positive. And also in the Staphylococcus section is the tetrads that we were talking about, Micrococcus luteus and roseus. So that's why we say it's Staphylococcus. But those ones are the catalase positives. Very good. Okay. This test is present for both major groups of bacteria. It looks for the presence of blank enzyme. A positive test is determined by a color change from orange to red, so bright red color. So yellowish to red. Bright red is really the color change that we're looking for. In this case, we are looking for pyr, pyridonylase, pyridonyl aminobiase, which is wrong enzyme. Pyr enzyme is much easier to say, and that's what I'd much rather you guys remember, because I don't want you guys to remember all the different variations and stuff. Basically, we're looking for the ability to break down pyr and break it into different little parts, and then those parts react with a chemical that we add into there, which turns bright, bright red when it's positive. So if it has that enzyme, it's able to break it and cleave pyr, then we're going to get a nice bright red color when we add the reagent to it afterwards. The reagent is aminocinnamaldehyde, dibenzoaminocinnamaldehyde. Okay, but you basically just call it pyr reagent. Okay, but that's the one that's for both of them. So we can use pyr for both catalase positive and catalase negative. That's the little broth test. And you should know how to be able to read those broth tests. They're pretty easy to read. Antibiotic for catalase test, and there's an antibiotic for catalase negative test. And I always forget these because I am terrible at remembering things. And if I can remember correctly, the one for the catalase negative organisms, so the ones that do not like a lot of oxygen, do you guys remember which antibiotic that is? SXT, very good. Bactrim is another way you could say it too, if you want to be fancy. And then for the catalase positive ones, it's a different antibiotic that has a tendency to do those. Sorry, I'm going to check to make sure if it's my lab assistant. No, it's not. Okay, great. Sometimes my lab assistant is doing things in the lab, and I have to check to make sure it's not them going, the lab's on fire. Sorry, I don't usually have my phone on. Okay. So what's the other antibiotic we use? So we use SXT for the catalase negative. For the catalase positive, we use novobiocin. Very good. Okay, and remember that each of these has their own measurement diameter when you look at them on the test to see if it's resistant or susceptible. You can't just go, oh, it's whatever number. You see a zone of inhibition period, you have to measure it. So measuring is important for these tests. The only slant test is for which media? BEA, and BEA stands for? What was that? No, that's PEA. This is the slant test, which is BEA, bile esculin agar. And so we're looking for the ability to process esculin into esculatin, and then it creates a nice dark brown coloration. You see dark brown coloration is positive, and if you don't see a dark brown coloration, it's negative. Okay. Blood agar is a media that gives you 3 possible results. And I know you guys have done this enough to know what this is. What are the 3 possible results for blood agar? Alpha, beta, gamma, or non-hemolytic. And you do need to be able to read with those. The one that trips people up is the beta versus the alpha. Alpha is just kind of like a dark coloration. Beta is like perfectly clear see-through. So if you can see your hands through the plate, that would be considered a beta hemolytic. It's complete lysis. Okay, does anybody have any questions on that? I may have skipped 1 or 2. Yes? 3 and 4, did I skip those? I'm sorry. Okay, blank test looks for the ability of organisms to break down nucleic acids, which is our DNase test, right? Oh, and I do remember you guys kind of ran into some problems with the DNase test. It looks for the presence of DNase, which is in the name, which is an enzyme that breaks down DNA molecules. So if you have the ability to do that, a positive test will create a clear zone, and a negative test, the entire plate will be cloudy. And this one you guys have to remember is one that we add a chemical to, which is hydrochloric acid. Hydrochloric acid causes nucleotides, DNA, to precipitate under acid conditions, and it becomes cloudy. So it makes the media cloudy. But if you have an enzyme that's been eating up all the DNA in the area right around the bacteria is going to be clear. It's not going to get cloudy at all because there's no nucleotides left. The bacteria has eaten them all up, used them for itself, so it's going to create a clear zone. I know that one kind of trips people up because sometimes we have clear zones and sometimes we don't. This is one where it's, if you have a clear zone, that means the bacteria made an enzyme and it destroyed the stuff around it. Yes? Right, okay, very good. Good question. So a positive test, so the negative test would be cloudy all the way through. And the reason why it would be negative all the way through is that means that there is DNA that was still surrounding this, and then when you added in the hydrochloric acid, it then made the entire plate cloudy, right? So we get the entire thing cloudy. But if you have a positive test, the bacteria is going to create an area around it where it destroyed all the nucleotides around it, and then the outside area, outside of this zone, is going to change to that cloudy color from the hydrochloric acid. But this area right around it will remain clear because the bacteria destroyed all the nucleotides there. So you're going to get a small little area that's clear, and everything around it is going to be cloudy. Okay, make sense? Yes? Good? Clear? Good? The clear zone will be in that streak, but it'll also be around because the enzymes spread outwards. So bacteria is just kind of dumping enzymes out into the nature, and so it's going to create a clear zone around the bacteria. So like a little halo effect, effectively. Does that make sense? Yeah, absolutely. Good question. Good stuff. Okay. All right. Negative tests. I think this one's not quite as helpful because it includes one of the things, the old style test that we did, which is not the style of test that we will be doing. But it's still the same fundamental procedures, which is we're going to be using those test strips that we used before. For the actual exam, I will provide you guys example test strips that will have color changes in them. But I'll also give you guys the color change card that tells you what every test is and what the colors should look like for positive, negative. So you still have to interpret them, but you'll have the card with you. And then you'll also get a chart that looks somewhat like this one right here. This guy right here, but with the correct tests on there. It's going to be a little squished down. So you guys will get another one, another test available for that so that you guys can look at that. And that one, there are only a couple of tests that are really important. Sorry, I shouldn't say that. All the tests are important, but you should be able to quickly recognize a couple of tests. I'm going to show you guys the cheat method of doing this. The very first one is the TSI slant for the gram negatives. That tells you two really important things. If it produces hydrogen sulfide. Hydrogen sulfide, there's only 3 hydrogen sulfide producers. That's Citrobacter freeundii, Proteus vulgaris, and Proteus mirabilis. So if you get black precipitate, and so when I say black precipitate, it's very black. It blocks out all the light coming through that slant on the bottom, then that is a positive. And that's an easy test because once you did that, you only have 3 organisms to choose from. So now all the other tests are just which of those 3. And that's a really immutable test. You can't really just get around that test. The other one for the TSI is whether it ferments glucose or lactose and sucrose. If it only ferments glucose, there's only a couple of organisms that only ferment glucose. One of them is Proteus mirabilis. I think the other one's Morganella. That's only 2 organisms. So that cuts your organism choices down by quite a bit. So those are really important tests. As far as the test strip, the ones that are really important are things like urease, the TET and lipase, and malonate are really kind of important. With those test strips, there are things that you have to add after the fact. So remember, you're supposed to read the test strip first, and then you're supposed to add in reagent to 4 of the wells. The first 3 wells are Remel reagent, and that's that The last three minus one, so not the Odonatol and other one. It's, what is it? Proline, GGT, and Pyre. And so you add the Remel reagent to those three. You're looking for color change for those three. The last one, Odonatol, also has a color test change to it that you read first, and then you add indole reagent and look for a secondary color change. I'll mark on the paper what that secondary color change is, so what the first one is, and then you'll have to interpret whether or not, when the indole's added, if it changes color or not. So just keep that in mind. But that one's not as important of a test. It's like low on the end of importance. Okay? Does anybody have any questions on the gram negatives? Yes, Proteus vulgaris, Proteus mirabilis, and Citrobacter freundii are the three H2S producers that we have in lab. And the glucose-only organisms, I believe, are Proteus mirabilis and Morganella morgani, I think, are the only two glucose-only ones. Sorry, did somebody else have a question? Yes. It's like 1 minute and 20 seconds. Yeah. Okay, but here's the thing. I know. You might be like, Josh, that's not enough time. I agree, it's not enough time. That's why we give you guys time at the end of this to go back and look at these. And we're going to have multiple stations of these identifications ones up so that you guys can look at those multiple ones so you're not creating a big line. The way I would go about it, and that I found students have had the most success, is they go, oh, hey, I can't interpret this right away. One of the things that they'll do is they'll just mark on their papers, you know, what the positive and negative bits are in a line, especially with the gram-negative test, because there's like a whole bunch of them. And that works for students. Most people can do the gram-positives really fast, so they don't necessarily need that. And then the second time around they go, they'll be like, oh, I can be able to figure out what it is off this chart. But those important ones that I said at the very beginning, where I said TSI, that's going to be kind of like your first easy hurdle to be able to identify things quickly. So I would definitely be able to go look at that and go, oh, that means it can't be, you know, if it's not H2S positive, it can't be those three. So those three are out. But if it is H2S positive, then it's those three. So it's kind of like cutting down the field of choices by a lot really quickly. Okay, good question. All right, anybody else have any questions about the gram negatives before I move on? Oh, that one, ignore. That's for the old testing. I forgot and did not update this. I'll try to put an updated one here on Canvas because I forgot that this is the old test style. We switched tests last semester and I didn't, or from last semester, so I didn't update it. Bioinformatics, two major forms of sequences are FASTA, which is just a little greater than sign, and then a title and a new line with all of your sequence. And the other one's GenBank, and GenBank is the one that has the huge amount of information about the gene. And then at the bottom, it has kind of everything broken up into little groups of 10 with numbered lines. And so you can figure out where you are in your gene really quickly with GenBank, but FASTA is much easier to type out. So if you have to like kind of make up a bunch of names and stuff, FASTA is the easy one. But you guys should be able to look at something and go, oh yeah, this is FASTA format, or oh yeah, this is GenBank format. And if we were like, hey, write a FASTA format, you should be able to do that because that one's really easy. I wouldn't make you guys write GenBank because that takes like an hour for humans to make. Bacterial identification often uses which gene? It is a universal gene that all bacteria have. It is 16S ribosome. And there's actually a specific region that we use, but you can actually use the entire thing. We don't usually do that because the sequencing is too long, but yeah, we want that little tiny sliver. And that one's conserved in that all bacteria generally have, so we use universal primers for that region. But basically, we look in that 16S region, it gives us a really good indication of what bacteria are. Most bacteria have a unique version of that, but they're all very, very similar. Some organisms have nearly identical ones, so Bacillus genus is really obnoxious because they all have the exact same 16S sequence. So you can't use 16S for everything, but it is a good landing, it's a good jumping off point. Okay. To run PCR, so universal primers, universal set of primers in order to run PCR. The website is NCBI, so that's the one that hosts BLAST. And I'm going to stress this because everybody messes this up on the exam. BLAST, basic, local, alignment, search tool. That's the acronym for BLAST. Make sure you guys know that. That's important. I know it's easy to shove in other letters in there, but don't. Make sure you guys know what that stands for. Okay. Completely synthetic PCR and gels are referred to as in silico. So when we do it in organism, it's called in situ. When we do it outside of an organism, it's in vitro. If we do it inside of a system, it's in vivo, blah, 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 blah. Okay, so in silico means it's in a computer. We do it digitally. We predict what it's going to be. Utilize predictions in a sequence where blank enzymes can cut DNA in specific location. So we can actually use little bits of enzymes, so things like EcoR1 or what have you. These are nucleic acid cutting enzymes, and they only cut in a specific location. So if you're like not exactly sure what your sequence is, you can actually use those enzymes to kind of cut things, and then by the fragment pattern, you can identify where it is or where your cutting sequence is. So it's actually a very handy tool. It's not used quite as much because just straight sequencing is way cheaper now than it used to be. We used to have to rely on those enzymes quite a bit more than we do currently. The two major databases we used in lab were, does anybody know? What was that? Ooh, very close. So those, BLAST-N and BLAST-P, are actually the algorithms we use. The database is what we're searching. In this case, we use non-redundant database. So non-redundant, basically say it's just one more or less copy of each organism. And then we can break it down to specific, and we can say, hey, we'd like you to look at only like the ref-seq. So we say, this is the reference sequence. So we say this is the canonical sequence for all Homo sapiens. They've all got the same gene here with this sequence, which is of course not true, but we say that's the reference sequence. So, and I believe that's the two that we used. What was that? Ref-seq, yeah. Reference sequence. So non-redundant database reference sequence. Okay, how are we doing on time? I don't want to go over too much. Okay, we're not over yet. Hold on. Okay, we're almost done. All right. PCR. PCR stands for what? Polymerase chain reaction. Very good. Hopefully you guys don't say it dumb like me where you say PCR reaction, because I know that's redundant and dumb. So please don't say it like that. Each reaction contains an enzyme. It's a really important enzyme. Which enzyme is it? Taq polymerase. It could just be a regular polymerase, but we all use Taq now because Taq can withstand those ultra high temperatures that you need for normal polymerization in a machine. Okay. And short DNA sequences that are called primers. Primers are very important because you need to give something for the DNA polymerase to start on. If you don't have the primers, the DNA polymerase just goes, I can't do anything. It just wanders off and just cannot latch on. It needs double-stranded DNA to do its job, and that's the reason why you need primers. In nature, we make our own kind of short sequences that the DNA polymerase can run on. In this case, we need primers, and this is the reason why we kind of have to have an idea of what's in that gene to be able to make primers. In this case, we use the 16S ribosomal primers because they're universal. Most bacteria, almost all bacteria, have them, and that sequence that we use for them are very, very, very conserved. You can't mess with as much in those regions, so those 16S primers are very, very good for us. We used to make a copy of the template DNA. So make a copy of the template. The machine that does this is called a what? Thermocycler, or PCR machine if you are lazy like me. Now, thermocycler is what it's called. It increases and decreases temperature in a consistent manner. And they can actually do a bunch of really crazy things like have gradients of temperature. That's especially handy if you don't know if your primer works at a certain temperature or not. So you can actually create a gradient to make the test out primers and stuff. It's very neat, very handy. It starts with a phase of high temperature to denature the what? Template DNA, yeah. So the very first thing you do, the template DNA, which is often genomic DNA, it's really stuck together, and so you have to have a very initial long cycle to denature it, to get everything separated out so you have single-stranded DNA everywhere, and that tends to happen about 95 degrees Celsius. So we want to have this really high temperature, so it spreads out. And typically we start, the very first part of this is going to be 5 minutes long the first time we run it through, and then after that it's about 30 seconds long. So the reason why we do it at the very first is because we're trying to get that genomic DNA apart, and genomic DNA is really sticky, so you need to give it enough time to fully pull apart. Okay, a series of cycles are repeated at various temperatures. These cycles contain 3 phases. Those 3 phases are denaturation, annealing, and extension or elongation. Extension and elongation, same thing. And you should know the temperatures of those. So denaturation, 95 degrees. Annealing is 55 to 65 degrees Celsius. And lastly, extension is about 72 to 75. 72 is kind of the standard one. Sorry, what was that? Elongation is about 72 to 75. 72 is like the default number, yeah. For a long time, I think is what I intended to put there. So it's about 5 minutes of time is what we actually do, so a long time. So that doesn't seem like a long time, but each of these cycles is only like 30 seconds a piece. So it's not that long. Okay, we are close to the end. Let's see here. What did we leave off on? 3, 4. After the PCR products run through a gel, agarose gel, with electrical current to move the DNA from the negative to positive charge. So DNA has a slight negative charge. Like charges repulse each other, so the negative and the negative will be pushed away towards the positive, and so it'll run that direction of the gel. There's a dye that binds to DNA only in the gel, which is super handy. The reason why that's super handy is because that means that the only thing that glows is DNA that's bound to that dye. So the dye only glows when it's bound to DNA, and that makes sure that you only have glowing bands where DNA is. Double-stranded DNA specifically. There's other dyes that combine single-stranded nucleotides, but we don't need to worry about that. Okay. And reacts with UV light or blue light. Both actually are true. In our case, we use blue light, but you could also use UV light. And you guys didn't run into this, but the last class, the 1102s, the transilluminator broke. So we had to use UV lights to do it, those are not transilluminators that we have. So you can use both UV light and blue light. So the ones that we use, blue light. But both answers are correct, UV or blue. In order to determine the size, typically in lane 1 and 10, we put a DNA ladder. So it's a set of fragments, and we can tell how big those fragments are. Some parts of the ladder are highly, a little bit brighter. That way you can go, oh yeah, that's the 1,000 marker, or that's the 1,500 marker, that's the 10,000 marker. In order to make it easier to add DNA to the gel is mixed with loading dye, 6X loading dye in this case. 6X is just heavy and it sinks really well for us, and so it kind of just pulls everything down so we don't have our sample just disappear into the water. It'll run through the gel in specific sizes. So the 6X loading dye has, in this case, ours has 2, but some of them have 3, and those run at different sizes roughly. That way you can tell how far your DNA is run through the gel without being able to see the DNA, which is really handy. And your gel, which is made of agarose, dissolved in, you guys remember what we dissolved it in? What was it? TAE buffer, 1X TAE buffer, yep. So it's not just water, it's buffer. The reason why we use buffer is because you need the salts for several things, but one of them is so that it conducts electricity and it runs through there properly. All right, if you guys have any other questions, come up and see me. I'll run over some things with you guys. I'll put some more worksheet stuff on Canvas so you guys can study throughout the weekend and next week. I will see you guys next week. And if you guys are OLO students, remember to shoot me an email. Let me know about which setup day you guys want, and I will see you guys next week.